Bismillahirrahmanirrahim. We are going to start the chapter 11, the biological membrane and the transport. We are going to study this chapter from the Lehninger Principles of Biochemistry 6th edition, and that was published in 2013. Why we need to study about the biological membrane and the transport? Because the membrane define the external boundaries of the cell. We all know the membrane is present to define the external boundaries. They are also controlling the molecular trafficking. That's why we need to study about the biological membranes. In eukaryotic cells, they divide the internal space into the discrete compartments. We know that in eukaryotic cell, there are membrane-bound organelles, and they are all made from the membrane. That's why we need to study about the membranes to understand about the function of all those compartments, about all those organelles. They organize complex reaction sequences and the central to both biological energy conservation and the cell-to-cell communication. Membranes are also contributing to the cell-to-cell communication. They are important communication. If the compartments are not there, energy cannot be conserved within the cell. So compartments, the inner organelles of the cells, are important in the energy conservation. Okay. Once again, we are here. So we are going to first of all see the composition of the different membranes, biological membranes. For example, the plasma. Plasma membrane is containing the highest quantity, highest composition, highest percentage of the cholesterol, the plasma membrane. And we can see that much proportion of the phosphatidylcholine is present in the plasma membrane. And inner mitochondrial membrane, we can see, is having the highest concentration of the cardiolipin. And the phosphatidylethanolamine is present in that much proportion in the inner mitochondrial membrane. Still, the outer mitochondrial membrane is having the different composition of different elements in the outer mitochondrial membrane, and we can see it is even having the higher concentration of phosphatidylcholine as compared to the inner mitochondrial membrane. Lysosomal membrane is having that much composition of different components, and nuclear membrane and the rough endoplasmic reticulum and the Golgi apparatus is having that much proportion of different components, different types of lipids in the membrane. So if we see the human myelin sheath, it is containing almost 30% of the proteins and 30% of the phospholipids, and sterols are present at 19%. And what kind of sterols are present in the human myelin sheath? It is the cholesterol. And other lipids like galactolipids and plasmalogens are also present. And you can see in the yeast, 52% of yeast membrane is having the proteins and 7% only the phospholipid and the 4% sterols. Sterols are less in the yeast. And E. coli is not containing the sterols in its membrane. So this is one of the representative model of the cell membrane, and we have heard many times about the fluid mosaic model of the cell membrane, and different components are shown here in this picture. For example, the galactolipid have been shown. This is the carbohydrate component that is attached with the lipid, that is the galactolipid component of the membrane. And the next one, the sterols are embedded in the cell membrane. We can see different sterols are here. And one of the transmembrane protein have been shown here. Transmembrane protein that is spanning through the membrane. Transmembrane protein that is spanning through the membrane is also having some of the oligosaccharides attached to this protein, and that protein that is making the glycoprotein, because carbohydrate component is attached with this protein. So the next component we can see is the phospholipids are there on the inner leaflet of the lipid bilayer. And we can see some of the proteins are attached through the GPI anchor. Glycosyl phosphatidylinositol component is attaching some of the protein to the membrane. And this is the lipid bilayer. This is the labeling, and we can see some of the peripheral proteins are covalently linked to the lipid, some peripheral proteins, and some peripheral proteins are attached loosely to the membrane sometimes because of some charge, they are detached because of some function, they are attached, and sometimes detached when they are not required. So these are the components of the cell membrane. So we can see this is monoacylglycerol and this is diacylglycerol. This is one fatty acid containing monoacylglycerol, and it is two fatty acid containing. Because of the composition of the acylglycerol, it can make different shapes. For example, the monoacylglycerol can make the micelles. Monoacylglycerol can make the micelles. A diacylglycerol cannot make the micelle, but the diacylglycerol, two fatty acid containing, it can make the lipid bilayer. It can make the lipid bilayer, or it can make the vesicles. You can see the figure, the shape of the lipid bilayer. Okay, we are again here. So I have taken this table for you from 10th chapter to let you know about the structure of the different phospholipids. You can see two fatty acids. Two fatty acids are joined to the two of the hydroxyl group of the glycerol. The red highlighted is the glycerol, and the yellow highlighted is the fatty acids. And this is diacyl glycerol, and it is having the phosphate group attached to third of its hydroxyl group, and there is a possibility of attaching one X group to the phosphate group. If this X group is hydrogen, this molecule, this phospholipid will be called the phosphatidic acid. These are all classes of phospholipid. These are all classes of phospholipids. If the ethanolamine is attached in place of X, this is called phosphatidylethanolamin. If choline is attached, this is called the phosphatidyl If serine is present, this is phosphatidyl These are different phospholipids that are present in the lipid bilayer in the cell membrane. That's why I'm showing you the composition, the structure of those lipids. So once again, a lipid bilayer is the basic structural element of the membrane. A lipid bilayer is the basic element. Definitely sterols are present, definitely proteins are present, but the lipid bilayer is making the basic structure. So phosphatidylethanol is present almost 30% in the almost you can see 70% in the inner monolayer, and very less proportion is present in the outer monolayer of the lipid bilayer. In the same way, different proportion of different fatty acid is present from inner monolayer to the outer monolayer, and we can see the composition of each in the inner and the outer monolayer. So they are having a different composition. Okay, we are moving next. So this is the cell membrane. We can see this is the cell membrane, and this is the transport vesicle containing definitely membrane, and this is the Golgi, and this is transgolgi, and this is the Golgi, and we can see definitely here the sphingolipids plus cholesterols are present in the inner in the inner leaflet of the transport vesicle, but the outer one, you can see this one. This one kind of blue or magenta, maybe some color, has showing the phosphatidylethanolamin plus the phosphatidylserine that is present in the outer leaflet of the transport vesicle. And we can see Golgi is containing this much composition. Its inner part is containing predominantly the phosphatidylcholine plus sphingolipids plus cholesterol, and but the outer one is containing phosphatidylethanolamin plus the phosphatidylserine. So we are right now talking about the composition of the biological membrane. We have not studied anything in detail, but only the composition we are studying right now. Okay, so what kind of proteins are present in the lipid bilayer? This is lipid bilayer. This is biological membrane, right? Okay, so some peripheral proteins are present in the lipid bilayer that are attached to the other proteins embedded into the lipid bilayer because of the presence of the opposite charge. So these proteins can easily, which proteins? The peripheral proteins can easily be detached by the change of pH. If we talk about the M5 tropic proteins, they are sometimes attached to the membrane and sometimes are detached to the from the membrane depending upon their function. If we talk about the GPI anchored protein, glycosyl phosphatidylinositol is present here, and the proteins are attached through this GPI anchor to the plasma membrane. These proteins can be detached by the action of phospholipase C. So right now in this slide we are talking about the different proteins and their strengths to which they are attached to the lipid bilayer. And the last type of proteins that I'm showing you here is the transmembrane protein that is spanning through the lipid bilayer. So these proteins can be detached by the use of detergent, because detergent can dissolve the um, uh, phos- uh, the phospholipids, and this protein will become free. So this protein is also tightly bound. This is tightly bound. This is very loosely bound, and this is even the loosely bound. The that is detached because of the change in the pH. Which protein? The peripheral protein. Okay, we can see this transmembrane protein once again. This is having one transmembrane domain. It is spanning through the membrane only one time. So that is one single transmembrane protein. So we can see these are the amino acids: leucine, serine, threonine, glutamic acid, valine, alanine, methionine, histidine, threonine. So these are the different amino acids that have been written with 3-word abbreviation inside this picture. These are different amino acids. So whenever a protein need to be inside the plasma membrane, need to be passing through the lipid bilayer, it must have a domain that is containing the hydrophobic amino acid. We can see all these amino acids that are present in the protein that are in adjacent with the lipid bilayer are all the hydrophobic amino acids. But the hydrophobic all amino acids are outside the lipid bilayer. They cannot be inside the lipid bilayer because the hydrophilic amino acids are repelled by the hydrophobic environment. So throughout this Lehninger notebook, wherever this light color is shown, it is showing that these are hydrophilic. Light blue color is denoted to show the hydrophilic environment, but light yellow color throughout this Lehninger textbook is showing the hydrophobic moieties. Whenever there is a hydrophilic amino acid, this is actually driven away from the hydrophobic environment. Therefore, because of the presence of some of the hydrophilic amino acid, there is a bend inside the membrane. But still, the hydrophobic amino acids are keeping even this bend inside the lipid bilayer in the hydrophobic environment. So there are actually the 6 kinds of transmembrane proteins, the 6 kinds of membrane proteins. There are 6 kinds of membrane proteins. We talk about them. First type, this is the inside of the cell and this is the outside of the cell. First type of protein is having the amine terminal outside of the cell and carboxylic inside. This is type 1 protein, membrane protein. In type 2, amine group is inside the cell and carboxylic is outside. In type 3 protein, one single protein is spanning several times through the membrane. One single protein, one single molecule of the protein is spanning through the membrane several times. This is type 3 membrane protein. In the type 5, we will follow this numbering. In the type 5, different molecules of the same proteins are spanning through the membrane and they are falling next to each other. Different molecules of the same proteins are spanning through the membrane and they are next to each other. This is making the type 5 membrane protein. And the type 6, and this is type 4, may be written type 4 protein, is linked to the membrane through the GPI anchor, the type 4 protein. And the type 6 protein, it has been labeled in a way that is also spanning through the membrane and that is also attached through to the phospholipid by the covalent bond. This is also attached with the covalent bond, and this one protein, the class 6 protein, is also linked through the covalent bond. Next, we are going to see the sheep aquaporin. In the sheep cells, the water is transported inside. So the aquaporin, actually transporters are responsible to pump the water, to take the water inside the cells. So what is the sheep? This is the fatty acid of the phospholipid, and this is the head group. This is the head group of the phospholipid, you can see. And the dark blue color, is showing the protein. This is the protein, and these ones are the phospholipid. And you can see in the aquaporin, the phospholipids are actually circulating around the protein part of the aquaporin protein. But in another example, V-type ATPase, vacuolar type ATPase, the protein is capturing some of the phospholipid to perform its function. We will see the function in detail in next some slides. Okay. For example, we come across a protein like this is glycoporin, protein, and this is having from 1 to 130 amino acid. It is containing 130 amino acid, and this is the baseline. Above this baseline are those amino acid of this protein that are hydrophobic in nature, and beneath is the those amino acid that are hydrophilic in nature. And we can see this is very small domain. This is almost falling on the baseline, and these are all amino acids that are hydrophilic. And you can see we can see a bunch of amino acids that are hydrophobic in nature. Again, most of the amino acids are again in hydrophilic line. So this hydrophobic bunch of amino acid indicates that these must be inside the membrane. This domain, this bunch of amino acid must be inside the lipid bilayer. So it is having one transmembrane domain from this hydropathy index it is showing. We can see another example. The bacterial adoption protein is having 250, for example, amino acid. It is having one hydrophobic domain, 2, 3, 4, 5, 6, 7. It is having 7 hydrophobic domains. It indicates that it may have the 7 transmembrane domains. This protein, which protein? Bacterial adoption. So in this way, we can judge this protein is the transmembrane protein or not from the hydropathy index of each of the amino acids. If the hydrophobic amino acids are present in bunch in continuity to each other, that may be making the transmembrane domain the hydrophobic domain of that protein. We can come across another example. So whenever the amino acids of a transmembrane protein exist, hydrophobic amino acid will be falling inside the lipid bilayer, but the hydrophilic will be falling outside, of the lipid bilayer. And we can see at the edges the red one, and this golden one, yellow one or golden one, red one and golden one are tryptophan and tyrosine. Both these are having the benzene ring and bit hydrophobic. That's why they are falling at the edge of hydrophobic region of the lipid bilayer. But inside, in the center, in the middle, exactly inside the lipid bilayer are only the hydrophobic amino acids, and the charged amino acids are well away from the hydrophobic part. These are outside of the membrane. Who the charged residue, charged amino acids of these proteins. And these are the example of different proteins. You can see, you can read their names. So the F-type protein is having the beta barrel that is made from the several beta sheets. This protein is having the 2 pair of beta barrels, and this protein is having the 3 pairs of beta barrel that are making the composition of this protein. Okay, so some of the proteins are sometimes are attached through the membrane through an amino acid that is falling somewhere in between the protein. This is the protein part. This is the amine part, amino terminal of the protein. This is the carboxyl terminal, and that is attached to the lipid through this sulfur. Definitely it must be cysteine, because cysteine is having the sulfhydryl group, and the S is present on the edge, and that can make the bond. And this protein is making the bond with its with the fatty acid through its nitrogen, and that is the nitrogen of the terminal amino acid, and that is making again the bond with the through the sulfur with the fatty acid. And you can see this is the protein that is GPI anchored protein. This is glycosyl phosphatidylinositol that is making attachment of this protein with the lipid bilayer with the fatty acid of the lipid bilayer. Who is making the connection? Who is making the bridge? GPI is making the bridge. This is again, so what is the topology, how the fatty acids are behaving in the membrane. You can see here the liquid ordered state L naught of the phospholipids that are present in the lipid bilayer. We can see they are well arranged. When the heat is increased, they are bit disordered. This is bit disordered state. In this picture you can see they are bit intermingling. They are bit intermingling. So so there is a small difference of energy between the two states. We are moving first next. So under the different temperature conditions, so different proportions of different fatty acids are present. For example, the ratio of unsaturated to saturated fatty acids is decreased when the temperature is increased. Definitely at high temperature only the saturated will sustain. That's why ratio of the unsaturated to saturated is decreased under the high temperature.
Excuse me, sir.
जी जी बोलो.
सर लिक्विड ऑर्डर ये जो स्टेट है एल नोट से पिछली वाली स्लाइड वो लिक्विड डिसऑर्डर्ड स्टेट इसका थोड़ा बता दें.
अच्छा क्वेश्चन मैं बाद में बताऊंगा. मैं चूंकि रिकॉर्डिंग भी कर रहा हूं, मैं क्वेश्चन के आंसर बाद में दूंगा आपको. ठीक है, आप लिख लें क्वेश्चन और हम क्लास में फिजिकली इसको सारा डिस्कस करेंगे. आपने वरी नहीं करना. आई विल डिस्कस एवरीथिंग. इवन यू राइट दैट 100 क्वेश्चन, आई विल आंसर योर ऑल क्वेश्चons. Okay. Okay. So what is happening? We are now looking at a specific um, phospholipid. We are looking specifically at one phospholipid. We have colored this one, for example. So if this phospholipid can go into the other half of the leaflet of the lipid bilayer, this is called transbilayer diffusion. This is called transbilayer diffusion, and this diffusion actually is very slow process. Usually in the cell membrane, the transbilayer diffusion does not occur. It's very slow process, but the phospholipid can move next to next in the membrane, and this is called the lateral diffusion. This is easily possible, and this happens in the cell. So there are three transbilayer translocations that are catalyzed by the enzymes. One is the flipase. Flipase is transferring the phospholipid from one leaflet to another, and that is definitely energy consuming. Without energy, it's not possible. Usually, it does not occur. And another one is the flopase, that is shifting from inside to the outside the phospholipid from one leaflet to another leaflet. Who is causing this kind of translocation? The flopase. Another scramblase is shifting the phospholipid in this way in the presence of calcium, you can see. So different enzymes are required, otherwise this transbilayer diffusion is not easy, and this one is very easy. This may be the exam question in the objective type questions. So how can we prove that the lipid bilayer is a kind of fluid mosaic model? So from this experiment we can see actually we stain all the cells of the cell I would say stain the phospholipid of one cell, and then we impinged some laser light on one spot so that there is a bleaching at a specific spot on the lipid bilayer of this cell. This kind of bleaching can easily be seen by the microscope, but after some time, after impinging the laser, we let it grow for some time, and we can see easily that after some time the all the fatty acids started covering the bleached place. So it means they are moving from here one place to another, and they are filling the bleached space, and even bleached phospholipids have moved to other place, so that everywhere they are moving. And this experiment proves that the fluid mosaic model of the lipid bilayer. So if we want to see the movement of one phospholipid within one leaflet, so for example it is starting from here, it is moving here, moving here, moving, and sometimes it is a finish point, it is moving so much, it is so much moving. And this is the scale 0.1 micrometer, so you can guess how much distance one phospholipid is traveling. This is again So what is then holding the lipid bilayer? Because it is too much mosaic fatty acids, phospholipids are moving again and again. For example, this is one of the channel, and this is a glycoporin, and you can see the glycoporin is attached inside of the cell with the spectrin class of protein, and spectrins are holded by the ankyrin proteins, and different spectrin, you can see nets are joined by junctional complex actin proteins. Actin is one of the housekeeping gene, housekeeping protein, and this way from inside some of the support is given to the membrane through attachment with the proteins. That's why the cell is containing the stable lipid bilayer, though if it is fluid like moving capability inside. So these proteins are actually helping from inside. So we are now moving to another thing. For example, at some of the places, the membrane is having the specialized phospholipids. For example, sphingolipids are predominantly present at some of the places in the membrane, and these places are called the rafts. These places are called the rafts. You can see the cholesterol is there, GPI anchored proteins are there. So what is happening at the raft places? The caveolin protein combination. Caveolin protein combination. What we are going to study, actually, we are going to study about the invagination and some budding of the cell membrane so as to take something inside, or so as to excrete something outside of the cell. So we are going to study this kind of mechanism. That's why we are considering the raft structure of the cell membrane, having the sphingolipids and having the caveolin proteins. Actually, these caveolin proteins work in the form of two proteins work together in the form of dimer. They are attached to the membrane phospholipid through covalent bonds. These caveolin proteins. Let's see how they perform the action. So they are actually present in the form of, you can see here in the form of dimer, and each of component of the dimer is attached covalently to the fatty acid of the lipid bilayer, and they are pushing power, and this way several of the caveolin proteins are making this kind of bubble, you can see in the membrane. And this is electron micrograph showing different invaginations. This kind of invagination occurs, and something through this vesicle is taken by the cell. And who is performing this function? Caveolin proteins at a specific site of the lipid bilayer, at the specific site of the membrane, are performing this invagination for the cell. And you can see it has many applications, like you can see exocytosis is occurring through this process, like endocytosis is occurring through this process, like fusion is occurring through this process, and viral infection is also occurring by the invagination, and sperm is entering into the egg through this process. This is having different you can see applications are different functions who the caveolin protein by the invagination. So what is happening? The positive charge containing these proteins are being attached to the membrane, where they will cause the invagination, and sometimes they are proteins are attached covalently. They are attached covalently to the phospholipids, and they are causing the invagination in this way. We can see another example. For example, this is the neurotransmitter that is packed inside this vesicle. This vesicle now need to go inside the cell. This vesicle is containing the V snare protein, and the T snare proteins are present on the cell, in which it will go inside, and snap to 5 protein is also present on the cell surface, cell membrane, in which it will go. First of all, these snare T snare or V snare will intermingle, and they will make the coiling, and then the half fusion will occur, and then all the neurotransmitters are taken up by the
Excuse me,
new
sir.
cell.
बच्चे
Yes,
lobby में यहाँ पे enter करते हैं.
क्या करना?
सर बच्चों को enter करते हैं मीटिंग में.
अच्छा जी, वेट कर रहे हैं स्टूडेंट्स?
जी सर.
यस सर, लॉबी
कर
में
लिया।
काफी स्टूडेंट्स वेट कर रहे हैं।
अच्छा, मैंने एक्सेप्ट कर लिया सबको, ठीक है। So there are different types. So there are different types of transport. We will see simply here. Sometime simple diffusion is there. Simple diffusion occurs only down the concentration gradient. Down the concentration gradient means from higher concentration to lower concentration molecules are moving, but The condition is that they must not be charged. Charged molecules cannot move across the lipid bilayer. Charged molecules cannot move across the cell membrane. For example, DNA cannot enter into the cell membrane. Not even into the eukaryotic cells, not even across the prokaryotic cell membrane, the DNA can enter, the charged molecule can enter, charged big molecules, DNA, etc. But small molecules sometimes can diffuse, but they diffuse through the down the concentration gradient. Another one is the facilitated diffusion. This is also through the transporters. This is transporter, this is transporter, and this is the channel. This is the channel. So textbooks are showing specific, they are keeping specific like uh, specific shape for each of the transporter and the channel. Okay, this is facilitated diffusion. Next is the primary active transport, in which energy is required, ATP is required to transport. Usually this transport is against the concentration gradient from lower concentration to higher concentration. This kind of active transport occurs. Sometimes there is secondary active transport. For example, one molecule is transported again through by the use of energy, and the other molecule is facilitated because of the transport of the first molecule, because the first molecule was using the ATPs. Second molecule, transport of second molecule is called the secondary active transport. Next one is the ion channel. This is again maybe gated or by a ligand channel. This is down the electrochemical gradient, because we talked about the ions. So ions mean they are having the some charge. Sometimes ionophore, some hydrophobic things encapsulate some of the ions, and because of this hydrophobic environment, this ion enters through the cell membrane and then is released, and thus it enters into the cell. There are different conditions under which the transport can occur across the cell membrane. So we can see here C1 is greater here in the one compartment, C2 is less. Simple diffusion is going to make the concentration in both the compartments equal. We can see sometimes the charge ions are somewhere higher in concentration in one compartment. In another compartment there are positive charges, and after some time down the electrical gradient the charge will be balanced. So this kind of balancing of concentration and charges occurs through the membrane. Okay, but if there is a one molecule need to pass through the membrane without the presence of transporter, it must have that much, it must have surpassed that much high energy barrier to pass through the membrane. But if the transporters are present, very less energy is required for it to be inside the cell membrane, to be inside the cell. So transporters are energetically favorable for the transportation of the molecules. So there are two types. You can see ion channels. Ion channels have usually the one gate. Sometimes it's open, the molecules are passing through it, and sometimes it is closed and molecules are not passing through it. It is having one gate. But transporter, for simplicity, they are telling this is having two gate. Some one gate is opened and the molecules are entering, then the other gate is opened, then the molecules actually get inside. So it looks like they are controlling, the transporters are controlling the transportation throughout the path until they are inside. But the ion channels are having only simple open or close control. They are not having the very fine control, but the transporters do have the fine control, having the two gates. So we can see another example of one famous protein, that is GLUT1 transporter, sometimes GLUT1 transporter. This is GLUT1 to 12 are 12 types of transporters that are all meant for glucose, and they are present in different type of cell in our different organs. For example, this is GLUT1 transporter. It is having 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12 transmembrane domains it has. This is the amine terminal of this GLUT1 protein, and this is the carboxylic terminal, and this is the top view of this transporter. We will study next in next slides. For example, this has to transport, this GLUT transporter has to transport the glucose inside. So as we keep on increasing, if we keep on increasing the concentration of the glucose, the molecule will more and more will be passing through this GLUT1 transporter, GLUT. GLUT1 transporter. And same like in Michaelis-Menten equation, same like in enzyme kinetics, we talk about the Km value. Km is the Michaelis constant, because in this case that it is the transport that is happening, we defined another we name it as KT, the constant transporter. Not is this is Macaulay's constant, but this is transport constant, and half of its the value, the concentration of the glucose at which the velocity, the transportation of the molecules is half of its maximum value is called the transportation constant. Transport constant maybe it is. And if we draw the graph, same graph between the reciprocal of the substrate concentration and the reciprocal of the velocity, it we get a straight line. We get a straight line. So this is the GLUT1 transporter, and you can see this is the outside of the cell, this is inside of the cell. Glucose is entering somewhere here, it is being fit. Then the orientation of the GLUT transporter is changed in a way the molecule of the glucose is released inside the cell. This way the glucose is transported by the GLUT1 transporter. So we can see there are 12 types of GLUT transporters. We can see GLUT1 is ubiquitous, is present everywhere. It has a KT value of 3. If the KT transportation constant, if this value is very small, the transporter usually is saturated very early, but it can sense even the very small concentration, but it will be saturated quickly. And you can see GLUT4 transporter is very important, because the diabetes, type 2 diabetes, is because of the GLUT4 transporter. These transporter is present in muscle, fat, and heart cell. This is having the KT value of 5. So KT value 5 mean, so it is 5 millimolar concentration of glucose is going to increase the velocity of this transporter to half of its maximum value. So this is another chloride bicarbonate exchanger. And this is the red blood cells. This is the red blood cells. What is happening in the respiring tissues? For example, in our skeletal muscle, in all our respiring tissues, what is happening? Oxygen is converted into the carbon dioxide, and carbon dioxide is taken up by the red blood cells, and carbon dioxide after interacting with the water is making the bicarbonate ion, the hydrogen ion, and the chloride ion, and the bicarbonate ion are then released into the blood. Bicarbonate ions are released into the blood plasma. They are dissolved actually in the blood plasma, and through the blood they are moving. And what is happening when this kind of blood plasma is reaching in the lungs? In the lungs, the bicarbonate are again entering into the red blood cells, and bicarbonate is going to make again reversibly by the action of same enzyme, the carbonic anhydride, the carbon dioxide, and the water. And this carbon dioxide is then exhaled. We take the breath, and we remove the CO2. So this is the example. Insulin. Insulin is binding to the insulin receptor, and this insulin receptor is inhibiting the vesicles containing the glucose transporter to go to the cell membrane. This way, the less glucose transporters are reaching the cell membrane, and less glucose is absorbed by the cells, and blood is having the high glucose concentration, because it is not entering into the cell. So this is the mechanism of type 2 diabetes. If we keep on eating the high energy diet, high carb diet, more insulin will be released, and this hyperactivation of intracellular domain of the insulin receptor is going to inhibit the formation of GLUT4 transporter, and the person will be suffering from the type 2 diabetes. Okay, so there are three types has been shown here. One is the uniport, molecules are moving in one direction. There are symport, two molecules are moving in the same direction through one transporter. And this is the antiport, in which one molecule is entering and the other molecule is going out. These two fall under the co-transport class. Primary active transport, we can see here the energy ATP is required to transport the molecules through the membrane against the concentration gradient. And in the secondary active transport, this molecule is transported by the use of ATP, and the other molecule then is facilitated by this active transport. The other molecule is thus transported by the secondary active transport. So this is a P-type ATPase. This is a P-type ATPase that transport the calcium. They transport calcium. We see first of all its structure. It is having two major domains. One is transport domain and this one is S domain. This is T domain and this is S domain. T domain is darker in color and S domain is bit brighter color, and this is the A domain where it is. This is the A domain of this P-type transporter, and this is the P domain phosphorylation domain, and this is the N domain. This N domain binds to the ATP and the magnesium. ATP and the magnesium which the nucleotide domain is binding to the ATP and the magnesium, and this domain is then phosphorylated and the calcium is transported after the phosphorylation occurs and the ATP is being utilized. So you can see different formats of this P-type domains. This is the calcium pump is having this kind of format, and the sodium potassium pump and the hydrogen pump and the gastric hydrogen potassium pump is falling under the category of P-type ATPase pump because its name is containing the ATPase. It means it will hydrolyze the ATP. It will utilize the ATP. Okay. We can see here its cartoon picture. These are two calcium ions, and this is the N domain. This is step number 1. Now the magnesium. Okay, ATP comes in action. ATP binds here to the N-type domain, and this P domain is phosphorylated. You can see small p has been written here, and calcium gets inside, and next their change in shape occurs and the calcium is released. Previously this channel was closed, now this channel has been opened, and this way this cycle continues, and this P-type ATPase is pumping the calcium by the help of the ATP and by the help of magnesium ion. It needs magnesium ion, and even at the end the magnesium ion is released after the completion of one cycle. Okay. So what is happening next? P-type ATPase undergoes phosphorylation during the catalytic cycles. Let me take attendance one time. Wait. Okay. So P-type ATPase once again. So what they are doing. So you must memorize very useful information. Potassium concentration inside the cell is always very high. Is high, 140 millimolar, and the sodium concentration inside the cell is low, 12 millimolar. But outside the cell, in the extracellular fluid, the potassium concentration is very low. In the outside of the cell, sodium concentration is 145 millimolar. I usually use, when I work with the protein, I take the concentration of sodium ion, usually 135 millimolar in my buffer solution when I'm dealing with the protein, when I'm working on the protein. So I'm keeping the sodium concentration that is present in the extracellular fluid. Okay. So what is happening? The sodium actually is pumped outside. Sodium is pumped outside by the P-type ATPase, P-type transporters. P-type transporter is transporting the sodium outside of the cell. To compensate this, the potassium is coming inside. This way, the sodium and potassium concentration is maintained inside the cell. And this is again the V-type ATPase. This was P-type ATPase. These are, and now the V-type vacuolar type, and this is the F-type ATPases. So these are, you can see the V-type ATPases are utilizing ATP, but this is not shown here. But these are pumping the hydrogen from the cytosol to the lumen of the vacuole to keep the concentration of H positive ion inside the vacuole at some specific concentration. And in the, you can see the F-type ATPase. This is the F0 domain. This is the F1 domain, and this is again is utilizing the ATP to pump the protons from the cytosol inside the mitochondrial matrix. So these are this is V-type and this is P-type ATPase. This is again pumping. So this is very interesting. Another type of transporter. This is the ABC type of transporter. ABC. You can see this shape look like A. This shape look like A. Maybe that's why they have given the name ABC. ABC type of transporters. This is having two nucleotide binding domain. It is having, you can see, and this is a specific example of transporter. This is having again the 10 transmembrane domain. This is again 10 helices. You can see 10 helices each one, and 2 NBD domains, 2 NBD domain. And in this cartoon picture, we see really the function. So some molecule from the outside, some molecule from the outside, if it is entering into the ABC type of transporter, then the two ATPs are required. Two ATPs are required that bind to the NBD domains. NBD domains. After the ATPs are hydrolyzed, the molecule is released in the cytoplasm. Molecule is released in the cytoplasm by the use of two ATPs. This is the example of ABC type of transporters. So what is happening in the E. coli? The lactose is going inside by the potential gradient of the hydrogen that are pushed outside when the fuel is combusted, when the fuel is producing energy. If the cyanide is provided in that environment, the lactose concentration inside the cell will keep on falling because the hydrogen ion will not be pumped and no fuel will be combusted. So under the presence of cyanide, the efflux, the removal of the lactose inside the cell will keep on falling, though the lactose in the media was enough, because lactose will not enter. So in the media, the lactose is at high concentration, so its concentration will not be affected. So we can see again here, this is the p-type transporter again here. So you already know two of the domains that are present in the membrane, and this is the NBD domain, NBD1, NBD2, and this is the R domain. So this kind of ABC transporter, this transporter can function only when this R domain is phosphorylated. When this R domain is phosphorylated, this can function only in under the condition. This is the normal condition. This domain is phosphorylated, then only the two ATPs can come in action to bind to the NBD domains. And chloride can go outside of the cell. But if this domain is not phosphorylated, they will bind directly to the NBD domains, and there will be no place, there will be no chance for the ATPs to bind to the NBD domains, and there will be no open channel, and channel is closed. This kind of condition, when this amino acid is mutated and is not phosphorylated, the cystic fibrosis kind of diseases occur because of this kind of mutation in which it is not phosphorylated. Who? The R domain of ABC transporter. And you can see there are different hydrogen transporters. They are sometimes symport, they are sometimes antiport, and you can go through this table. These are very useful information. And you can see here once again what is happening. One carbohydrate molecule is coming into this transporter. This is the lactose transporter. And you can see these are the charged arginine, maybe these are green one, or some of the arginine red sugars, and the green one are, okay, the green ones are arginine. Because of the change in the charge of this arginine, these some of the amino acid present here, like glutamic acid, are repelled, and the channel is opened, and the lactose is transported inside because of the interaction between the glutamic acid and the arginine. Glutamic acid definitely is the acidic amino acid, and arginine is shown here somewhere basic, that is feeling repulsion from the glutamic acid, and the channel is opened for the entry of the lactose that has somewhere here initially was present outside. Okay, so this is very useful example. So glucose and sodium are co-transported inside the cells present in the small intestine, but through here the sodium is pumped outside of the cell through this ATPase class of transporters, and the glucose is entering into the bloodstream through the GLUT2 transporter. Intestinal cells are having the GLUT2 transporters, and because the sodium is pumped outside, the potassium enters into the cell and is having the high concentration of potassium inside the cell and having low concentration of sodium inside the cell. Because of this sodium potassium ATPase, sometimes we say sodium potassium pump, sometimes we say sodium potassium ATPase. So this is performing function to keep the concentration of sodium at lower level in the intestinal cells. So this is valinomycin, one of the potent antibiotic. This is one of the potent antibiotic. So what is happening? What is happening? The hydrophobic amino acid side chain are shown in yellow. Hydrophobic side chain are here, yellow, and you can see potassium ion are kept. Potassium ions are kept by this drug. So because this is having the hydrophobic moieties, this medicine, this drug, this cyclic drug can easily enter into the cell membrane. That result in the when the potassium ion, many potassium ion enter into the cell, the membrane ion gradient is disturbed. Membrane ion gradient, because of the presence, because of the entry of several potassium ions inside the microbe, the ion gradient is disturbed, the microbes are killed. So this way, this is the mechanism of the valinomycin, a potent antibiotic. So these are different aquaporins here that transport the water. You can see these aquaporin, read by yourself. So I let you know that this is the enzyme, this is the protein, this is the channel aquaporin, and there are two importance. One is this one. This alpha helix and this alpha helix are actually facilitating the transport of water molecule. They orient, these two green chains orient the water molecule in a way that they don't make the hydrogen bond, and they are transported through this aquaporin. So I think we end here. Okay, last one. Sometime if we want to know what is the potential, how much transportation a channel can perform, sometime we pinch from the membrane, the small piece of the membrane through this, you can see forceps, and we dip this forceps into a solution, and we attach this forceps through an electrical you can see pulse, and this electrical pulse is then applied through this small pinch of the membrane and directly to the solvent, and whatever the amount of current is passing through this ion channel gives the strength of this ion channel, how much it will transport, how much ions per second can pass through it. That can be calculated by the intensity, by the amount of current that can pass through this pinch and this circuit. So that's all. Thank you very much. तो जो क्वेश्चन आपके होंगे, ये मैं आपको पोटेशियम जो है चैनल, सो यू प्लीज रीड दिस पोटेशियम चैनल बाय योरसेल्फ, एंड दिस इज द एंड ऑफ चैप्टर, राइट? So we will discuss everything in the next class.